Now we go to the last one. Okay, we go to the last one. We are going to talk about urea formation. Just now we have the urea. Just now we have the apa? We have the ammonia, right? The ammonia will be converted into urea. So for the urea formation, we are going to learn into details lah. What is the urea cycle? How is the urea cycle? That at the end of the day, the ammonia will become the urea. Okay. Okay. Okay, so this is the urea molecule. Urea molecule, they have two amino groups. Okay. They have two amino groups. So one nitrogen atom comes from the ammonia just now. Through the process of deamination. Through the process of deamination, you guys remember it removes the amino group and then the amino group will plus with the H and becomes the ammonia. So one of the nitrogen atom comes from ammonia. And then the other amino group, they comes from the amino acid aspartate. Okay, so that's why in this formula we have the aspartate here as the donor of the one of the nitrogen group. Okay, and then we also have the ammonia here as the donor of the amino group for the urea. Okay, and then for us to make the urea, we also need carbon dioxide. You guys remember this one need to hafal. We need carbon dioxide. And then we need ATP. ATP means we need energy lah. Means we want to produce urea also we need energy. Okay. And then also we need water. Okay. We need water. So you guys remember all these components lah. Okay. And then the product is the urea itself. And then because we need the ATP, right? So ATP will be broken down. So if it's broken down, so it become ADP lah. And also P. And then we have the AMP. And then also we will produce the fumarate here. Okay, so this is the overall formula for the urea cycle. Okay, and then the formation of urea is a cyclic process. It's a cyclic process, kitaran. Why did we say it is kitaran? Because it is continuously we generate the ornithine. Okay, that's why it's a cyclic, because we are using the ornithine and then we will regenerate back the ornithine. Just like the Krebs cycle. Why is it that we say Krebs cycle? Because the Krebs cycle, we use the oxaloacetate, and then at the end of the cycle, the oxaloacetate will be reproduced. That's why it's called Krebs cycle. For the urea cycle, it's ornithine, regeneration of ornithine, involving the ornithine. Okay, so ornithine is thus regenerated and can be transported into mitochondria to initiate another round of urea cycle. So this urea cycle happening in the liver lah. Okay, happening in the liver. We have two locations in the liver. One is mitochondria of the liver and also the cytosol. Cytosol also known as the cytoplasm. Same thing. If in exam you want to say cytosol also okay. You want to say cytoplasm also okay. Correct. Okay. So there's two locations here. One is mitochondria, another one is cytosol. These are all in the liver. Okay, these are all in the liver. Okay, so we go to the, because they have 5 steps. If you see in this picture, we have step 1, step 2, step 3, step 4, and also step 5. Can. Okay, so we have 5 steps together. So now we look one by one. For step 1, condensation of ammonia. Just now we have the ammonia, right? The ammonia from the removal of the amino group of the amino acid during deamination, remember? Okay, so we have the ammonia, condensation of ammonia, and also the carbon dioxide. Okay, the carbon dioxide actually in the form of, it will be in the form of bicarbonate ion, HCO3 minus. Okay, and then it will form the carbonyl phosphate in the mitochondria. So if you see in this diagram, we have the bicarbonate here and also the ammonia here, but in this reaction they write it as ammonium ion, NH4+. Okay, correct also. Okay, so the ammonium here plus with the bicarbonate. Okay, and then it's also use ATP. You see, it's use ATP here, and then it becomes the carbonyl phosphate. The enzyme, this is optional lah, no need to remember lah. If you want to remember, can, but not so required lah. The most important thing is you remember the bahan, you remember the molecule involved. Ammonia, carbon dioxide or bicarbonate ion in the form of bicarbonate ion, ATP, and become the carbon monophosphate. Any question?
No.
Okay, so that's the first step. We go to the second step. Second step, we have the carbon monophosphate. We combine with the ornithine, and it will become the citrulline. Okay, and then this one happen in mitochondria, then the citrulline will be transported out of the mitochondria into the cytosol. So you see the picture here, we have the carbon monophosphate, will combine with the ornithine. Okay, will combine with the ornithine, and it will become the citrulline. Okay, and then once it becomes the citrulline, it will go out of the cytosol. This is mitochondria. Now they will be transported out of the mitochondria to the cytosol. Now citrulline already in the cytosol. Okay. Okay. So you see the formula here. Carbon monophosphate plus with the ornithine and become the citrulline. Okay. Okay. Next one, step 3. What happens to the citrulline? Citrulline is condensed with aspartate to produce arginosuccinate. This reaction requires ATP. Okay. The second amino group of urea is incorporated in this reaction. Okay. So you see here in this picture, we have the citrulline. The citrulline will will condense. We say condense. Will be condensed with the aspartate, and it becomes arginosuccinate. Okay. So just now we have the amino group. We have the amino group. Okay. This is the arginosuccinate. This is the structure of arginosuccinate. We have 2 amino group here. We see here 1 and 2 here, right? So one amino group comes from the ammonia, where the ammonia comes from the deamination just now, and then this now the next nitrogen or ammonia, the next amino group comes from the aspartate here. This is the aspartate. They have the amino group here. Okay, so they donate for the second amino group of the urea. Okay, so the formula citrulline plus the aspartate and then requires ATP. Okay, you see here it needs the ATP here. Okay, and then it becomes arginosuccinate. Okay, in this formula it's written as argininosuccinate. Okay, some book they use the word arginino. Okay, but some book they use arginosuccinate. I want you guys to use the word arginosuccinate. Okay, but some international book also use arginosuccinate, also correct. Okay, but you guys use this word arginosuccinate.
Okay, teacher.
Alright. Now what happens once we have the arginosuccinate? Step 4, the arginosuccinate will cleave. Cleave tu maksudnya dia akan breakdown lah. Cleave or breakdown into arginine and also fumarate. Okay, so this fumarate will be liberated. Will be liberated means will be released. Will be released from the will be released from the cell. Will be released from the cell or will be released from the cycle. Okay, and then they will link to the Krebs cycle later on. Okay, so if you see in this diagram, arginosuccinate now, they will be cleaved or broken down into fumarate and also arginine. Okay, fumarate will be released, arginine will be further used to produce the urea. Okay. Okay. So fumarate it is removed from the arginosuccinate, then will leave the nitrogen atom on the other product, which is arginine. Okay. So we see the step 5. Okay. Now the arginine will break down. Okay, the arginine. Last step, the arginine will then break down into ornithine and also urea. Now we have the urea already. Okay. So the arginine will cleave into ornithine and also urea. Now the urea. Now we have the urea already. Then the urea will be transported to will be released from the liver cell into the blood. Blood will transport the urea into to the kidney. The kidney will filter the blood and then will produce the urine. And then the urine will containing the urea will be excreted out from our body through urination. Okay. And then the ornithine here, what happened to the ornithine here? So the ornithine here then can be reused for the other step of urea cycle. So that's why we say here just now, initially we use the ornithine, right, to combine with the carbamate phosphate. Now we, at the end of the cycle, after step 5, the ornithine is regenerate back and be used for another cycle. Okay, so the ornithine is transported back into the mitochondria to initiate another round of urea cycle. Here, ornithine combined with carbamate phosphate and become the citrulline. Then the citrulline goes out from the mitochondria, go to the cytosol, and the cycle continues. And then carbon from fumarate also, just now we have the fumarate here released, right? Okay, so the carbon from fumarate may also re-enter the mitochondria and be recycled by enzymes in the Krebs cycles to oxaloacetate and ultimately to aspartate. Here, this is the aspartate. Okay. So actually, this fumarate at the end, they can be they can be re-enter the mitochondria and then they will be converted into aspartate and then be used back in the urea cycle. Okay. Okay. So this is the overall formula you can refer. Okay. If you see here, this diagram just want to show you the connection between the Krebs cycle, TCA cycle is the Krebs cycle, and also the urea cycle. Okay, so if you can see here, this fumarate just now, the urea cycle just now released the fumarate, right? So actually the fumarate, they can be converted into aspartate in the Krebs cycle. That's why we say the urea cycle connected to the Krebs cycle just now, okay, through the fumarate. So the fumarate actually, they can be converted into malate and then converted into oxaloacetate, and then can be converted into aspartate, and then the aspartate can be used back to form the arginosuccinate to join the urea cycle. Okay, so this is all the this diagram all about to show the connection between the urea cycle and also the Krebs cycle by this fumarate molecule. Okay, okay, and then this urea cycle you can see here, it will produce the urea here, and then we'll regenerate the ornithine, and then the ornithine will be used back. Okay. Okay, and this diagram want to show you the source of the nitrogen actually. Okay, if you see here, actually most of the nitrogen actually comes from the glutamic acid. Okay, comes from the glutamic acid. Glutamic acid also known as the glutamate lah. So this glutamic acid, they undergo the deamination. So this glutamic acid, they undergo the deamination and then they contribute through the deamination, the glutamic acid will remove the amino group and then it will produce the ammonia. So this is the first source of ammonia comes from the glutamic acid. And then another source, just a little bit to either acetate from acetate. Okay, and then another one, the source of ammonia is from this aspartate. Okay, actually that's all. Okay, that's all for the urea formation, and that's all for